benefit of the
Are those comments that he writes at home?
Or like other times, like he'll like write it his own and land it, and he'll be like, never mind. Like, where is this? Oh, I found it. Alright, thanks, bud.
Okay. Happy Friday. Okay, so the first thing I just wanted to remind everybody of, and I think most people are aware of this, but the first worksheet is due tonight. I know that the MO2 Dropbox had the wrong time on it for some reason. I don't know why that happened, but so I changed it. 11:59 tonight is the deadline, so some of you have already turned it in if you haven't, do that before then. If you need access to a scanner so that you can scan your lovely chromosomal drawings, the printers actually all around campus, but particularly in this building, all you have to do is swipe your card, hit the email button, and then hit send, and it'll scan as a PDF and send it to you in an email. So, yeah. So take advantage of that, use that, try that. Yeah. Okay, so sort of my plan for today is to cover a little bit of lecture and then give some time for questions. Overall, how is everybody feeling about mitosis and meiosis? Good, bad? Okay, give me some indication. Yeah. Okay. All right, so most people are feeling at least ish about it, so I'm going to let it rest for now, and then I can answer questions, or we can review it again before the first exam. Okay, so we will move into our inheritance stuff, and actually we're going to see meiosis again here, so we get another sort of opportunity to actually review it in this material. So I ended on Wednesday sort of talking about the early theories of inheritance. It wasn't until the early 1900s that we knew about chromosomes. It wasn't until the 1940s, 1950s that we knew DNA was our actual unit of inheritance, right? So these things seem kind of crazy, but 120 years ago, we really knew nothing about chromosomes and DNA and inheritance, so people were more or less sort of theorizing how these sort of things worked, right? And so they're kind of fun to look back on and think, like, to us it's like, wow, this is just kind of ridiculous because of all the stuff we know now, but at the time, this is what made sense to people. Okay. In the 1800s, we had Gregor Mendel. Mendel was going to be sort of our father of transmission or classical genetics. He gave us a lot of really important information when it came to how traits are passed from generation to generation. So his actual research was done between 1856 and 1868-ish. So about 12 or 13 years he actually did this research. And at that point, so in the 1860s, he stopped. He didn't die, he didn't decide science wasn't cool, but he was asked to take on sort of a different job. He was a monk, if you don't know. And so he kind of just moved on to other things and left his work unpublished for quite a while. Published poorly, I guess, for quite a while. So I know that everybody loves videos. This is a story about Gregor Mendel.
Oh, why, dear God, did you make it so complex to understand the offspring that result when there is sex? But there is one monk among us who can tell how it all works. We feel like jerks next to Gregor Mendel. The answer's in my garden where I planted different weeds and sprinkled pollen as I please dominate. Later on, the world was awed at what he learned from those bonds. But back then, no one heard of Gregor Mendel, but his God. Back then, no one heard of Gregor Mendel, but his God.
All right, so that's an introduction to Gregor Mendel. Okay, so Mendel actually did publish his work. Okay, so it was published in 1866. It wasn't until much, much later, so in the actual 1900s, so 40, 50 years later, that his work was actually sort of brought back into the light and was used to talk about how inheritance happened. Okay, so he did all this research, he published it, and then it sat without anybody sort of realizing what it was until we had 3 botanists, and they actually did this independent of each other, but they pulled his work and realized that it actually told us how genes are inherited. And it's because that's the title he used for his paper, for his publication. It is not a very eye-catching title. Right? How many people want to read experiments on plant hybrids as part of their... No. Could have titled it much better, and it may have given some indication as to what it actually was. So when you guys are titling your research projects, make them exciting titles. Okay. So most of you probably are familiar with Mendel and his work a little bit. Is that a fair assumption? Okay, good. So we're not working in the dark here. All right, so Gregor Mendel worked with pea plants. Okay, this is a very standard garden pea variety. He actually got pretty fortunate by picking the pea plant because there are many traits and the modes of inheritance are sort of easy to track. We'll see next week, we're going to start talking about non-Mendelian inheritance and how easy it would have been for him to select a plant or traits that did not follow our Mendelian inheritance laws, which would have made, well, we wouldn't have Mendelian inheritance laws then, right? Because he would have known what the heck he was looking at. Peas happen to just be good model organisms. They do have very distinct and sort of easy to differentiate features. Okay, so they're he looked at height, pod structure, coloration, flower color. I'll show you the 7 traits he looked at, but they're really easy to see. Okay, so very easy to distinguish. He was able to work with true breeding strains, which I'll work with, which I'll talk about in a second. Okay, so these are the 7 characteristics that he actually looked at. Right, so seed color. It's very easy to see a yellow seed compared to a green seed. I think even if you're, actually I'm not sure if you're colorblind, maybe not. Let's assume he wasn't. The pea shape. Okay, so some peas are just very round and plump, and then some are sort of wrinkly, easy to see. The seed coat color was either gray, so it's this piece, or it was white. Pod color, green or yellow. Pod shape, again, either sort of fat and plump or sort of constricted around the seeds. Where the flowers were located, and then how tall they were. So it was very easy for him to say, I'm going to look at this particular trait. I'm going to breed this really short plant with this really tall plant, and then I'm going to count the offspring and whether they are tall or short or in between. Okay, so before we get into sort of the experiments that Mendel did, I just wanted to run through some terms. Most of these terms we've talked about, I think I've used all of them, but I don't think I've defined all of them quite yet. So everybody is comfortable with what a gene is? Okay. So what do genes code for? I know it's Friday, but traits. How are traits determined, or what part of a cell determines a trait? We're going backwards. All right, we start with DNA. DNA contains genes.
which are used to make amino acids, which make proteins.
Yes. Okay, so these genes code for particular proteins. The proteins that are present in a cell give that cell its characteristics, right? So when we say that a gene is an inherited factor that helps determine a characteristic, what we're saying is a gene is a piece of DNA that's passed from parent to offspring. Within a cell, the presence of particular genes code for proteins which give a cell its characteristics. So again, I will remind you that every cell in your body contains every gene in your genome. You have 46 chromosomes in every single one of your cells, but we only express the genes that code for proteins that make that particular cell function. There? I'll have to start bringing caffeine pills or something for you guys on Fridays. Yes. I feel like that might not be it. Okay, so then alleles are going to be variants of a gene. Here we have, what is this representing? Chromosome. Down here, the black and white are alleles of a gene, right? So this location on this chromosome is a particular gene. Those are the same gene, but different alleles to different variants. Okay. Locus is going to be where on a chromosome these genes are found. So I don't know that I mentioned this, but chromosomes we know are one big long string of DNA, right? Yes, okay. DNA is going to be nucleotides, A's, C's, T's, and G's all strung together. We can start at the top of the chromosome and count how many nucleotides there are. So A, C, T, G, 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 whatever, all the way down. We then can say at position 100,053 is the start of gene R, okay? And that tells us the locus. That should be consistent from person to person, or from individual to individual. Okay, so locus is simply position on a chromosome, like physical position on a chromosome. All right, genotype and phenotype we've talked about, but I don't know that we've specifically defined those. So genotype is going to be the set of alleles that an individual has at a particular gene or at a particular locus. So what is the genotype of this individual for gene R? Big R, little r. Okay. They could also be little r, big R, big R. Phenotype then is how this trait looks. Okay, so in this case, we have a big R and a little r. What's the difference between a big R and a little r? Right, so one of these is going to be dominant and one is going to be recessive in the classic Mendelian system. Which one is dominant? Yeah, we use capital letters to indicate dominance. So in this case, we would say the big R allele is dominant over the little r allele. The way that we should really define this is by looking at a heterozygote. If you look at a heterozygote individual and you look at their phenotype, you can then determine which of these alleles is actually dominant. Okay, so let's just say that this codes for red eyes is dominant over white eyes. If we're looking at a heterozygote, they have a copy of big R and a copy of little r. If they have red eyes, then we know that this big R allele is dominant. And the big and little is really just arbitrary. Little r would be recessive. Okay, so heterozygotes are going to be any individual that has two different alleles. So anytime you see hetero, it means sort of opposites or different. A homozygote then would be any individual that has two copies of the same allele. So either big R, or little r. Most of the time, I will specify, or your book will specify, homozygote dominant, which would be two big R's, or homozygote recessive, which would be two little r's. Okay, and then a characteristic or a character is just a particular trait or feature that we're looking at. So it would be like hair color, eye color, height, whatever trait you want to base it. Be comfortable with vocab. Okay. I will tell you that on exams, I think the biggest issue is actually reading the question and looking at the vocab that's there and then answering based on that. Not because I'm trying to be tricky, but just because there's a lot of terms and it can get, so that's why I spend time going over these terms. All right, so we are going to talk a lot about true breeders in this chapter and in future chapters. So before we move on, I also wanted to define what a true breeding individual or a true breeding line or lineage is. And so what a true breeding lineage is going to be an individual or sort of a family of individuals that is homozygous for a particular trait. So when we say that Mendel took two true breeding plants and then crossed them, we would be talking about plants that are homozygous for the trait that he's studying. Right, so if he took two true breeding plants, one that was tall and one that was short, and crossed them, one has two dominant alleles, one has two recessive alleles. So here's my example of this magnificently mean creature. Mean. Siamese are notorious for being mean. Yeah. I'm not all of them are, that's clearly a stereotype of Siamese cats. So there are a lot of really distinct features in a Siamese that make them look much different from your domestic short hair, which would sort of be what most people picture as their cat, right? Sort of that banded, darker coloration, kind of a consistent coloration. But here we have a longer face, we have blue eyes, we have these big ears, and then we have point coloration, which would be areas on the face, the tail, and sort of the feet that are darker. The way that we get a true breeding individual like this is we essentially take a normal cat, we look for traits that are kind of like what we want, and we breed it with another animal that looks like that. And then in those offspring, we pick the two that are closer to what we want and breed them together over and over and over and over again until we get sort of this very weird, genetically weird individual who has a lot of recessive traits.
Is that true for all breeds of like dog or cats?
Yes. Yeah, so that would be an example of artificial breeding. It's how we get all of our production animals, so like cows, pigs, chickens. They don't look anything like what a cow, has anybody seen like what the ancestor for cows looks like? It doesn't look anything like an actual cow. It looks like a buffalo almost, right? And then it's like a cross between a buffalo and a deer, and then we bred it into Holsteins, right? Or Angus or whatever it was. And so yeah, that's how we breed for our companion animals. How many people love like French bulldogs and pugs? All those dogs that sound like they're constantly underwater when they're breathing. Yeah. So those are called brachycephalic dogs. Very short faces. That's not a normal trait. That's very recessive. And actually, for all of you bulldog lovers out there, most of them can't give birth naturally, so they have to be C-sectioned when they breed them.
The same for Persian cats and stuff?
Not as extreme. When you think about a dog's face, like think about a wolf, right? They have a very long face. A wild cat still has a fairly short face. So like, yes but no. Yeah, so those are all recessive traits. If you put a pug out in the wild, let's just say they would make a predator very bold. All right, so I am honestly a little creeped out by short-faced dogs. Can't listen to them; it makes me uncomfortable. Okay. All right, so we are going to walk through basically two types of experiments that Mendel went through. Mendel was really, really excellent in what he did because he was thorough in his records, and he repeated these crosses sort of over and over again and just had a huge amount of data, right? And he recorded it very nicely for us, so we can go back and look at this. We're going to start with monohybrid crosses. Monohybrid crosses are going to be the sort of most simplistic version of transmission genetics. It's where we're just looking at one trait that has two states. Okay, so again, tall and short, yellow or green, wrinkled or round. Okay, just one or the other, just one characteristic. And so this is probably going to seem pretty, you probably have done this before. Okay, so a monohybrid cross is going to be a cross between two parents that looks at just one characteristic that has two states. And this might seem really basic, but the outcome that Mendel found was really, really important. So what he found was that one trait is actually determined by two genetic factors. Remember that when Mendel was doing this work, they didn't know what chromosomes were. They didn't know what genes were. So he uses terms like genetic factor. What could you replace that word with that we know already? So one trait. Yeah, so one trait is encoded by two alleles. Everybody comfortable with that? I hope so, right? These two genetic factors, which we've now replaced with the term alleles, are going to separate from each other when gametes are formed. We literally just saw that. When you make a gamete, so when you make an egg or a sperm, your two parental alleles duplicate and then divide so that each of the four daughter cells gets one of them. They're carried on chromosomes, but we're looking at individual alleles here. Okay, another piece that we have implied but not explicitly said is that these two factors separate with equal probability in the gametes. So if we want to think about this on sort of a really general level, I think it's easiest to think about males with the X chromosome and the Y chromosome. Right, so during meiosis, again, they replicate and then they divide. So what proportion of the sperm are going to carry X chromosomes? Half, right? You have two chromosomes, they duplicate, and then they divide into daughter cells. So you get, out of 4 sperm, 2 are going to have X chromosomes, 2 are going to have Y chromosomes. The same thing is true with all of your other alleles. Okay? They divide and then split. And so if you, regardless of what your allelic combination is, your gametes should have equal probability of being or containing any of them. Okay. All right. All right, and then this also allowed him to demonstrate what we already know as our concept of dominant and recessive traits. So the last piece that I need to tell you before we actually look at how he laid these experiments out is that pea plants have the ability to be both self-fertilizing and cross-fertilizing. So in self-fertilization, we have, why bother? Also, where's the other one of these things? Somebody steal it? Wasn't me? All right, so in self-fertilization, we have egg and sperm coming from the same parent. It is naturally occurring in peas. I want to make it very clear the difference between asexual reproduction and self-fertilization. Are those the same things? No. So asexual reproduction is basically the process of mitosis that results in 2 individuals. In asexual reproduction, every offspring is identical to the parent. In self-fertilization, we actually have 2 gametes. So we do have an egg and we have a sperm. Is every offspring going to be identical to the parent? No. Right, so if this self-fertilizing parent is a heterozygote, they could have offspring that are dominant, homozygote dominant, heterozygote, and homozygote recessive. Okay, so it's not the same thing as asexual reproduction. Okay, so for cross-fertilization, then we have egg and sperm coming from different parents. Right, so we have two parents that are contributing to the offspring instead of just one. So Mendel was able to manipulate the plants. Yeah, so he was able to actually manipulate the plants at different times in the experiment. So when he wanted to do cross-fertilization, so he wanted egg and sperm to only come from two parents instead of self-fertilization, he would actually go into the flowers before they bloomed, cut off some of the reproductive organs so that these anthers could not, is that right? Yeah, could not fertilize this plant's stigma. Right, so that these plants have both male and female, and I don't want to get into plant anatomy because I will mess it up. But this plant now only has one set of reproductive organs, so it cannot fertilize itself. So he was able to sort of manipulate that. Okay, so here is the process that Mendel used for monohybrid crosses. So again, this is just looking at one trait. He starts always with two true breeding plants. And so our example here, I realize that this is kind of small, but our example here is with a tall plant and a dwarf plant. These are the parental generation, so this would be P is how we would term this particular group. P stands for parental. So this is what the plants look like. This is their genotypes, right? They're true breeding, so we know that they are homozygous dominant, homozygous recessive. When he crossed them, so here would be where he did cross-fertilization, right? Cross these two. The F1 generation looked like this. All right, so F1 stands for first filial. This. First filial. It just means the first generation or the first sort of group of offspring. So if we do our Punnett square, and yes, I'm going to make you tell me what this Punnett square looks like. All right, so we have, do this part, we have one parent and then we have the other parent. So what do our offspring look like? Yeah, so they're all heterozygotes. And so this is their genotypes, what are their phenotypes? They're all tall. Okay. So in our F1 generation, they're all tall. The next part of Mendel's experiment was allowing the F1 generation to self-fertilize. Okay, so parental generation cross-fertilizes. F1 self-fertilizes. So if every one of our F1 generation is heterozygote, our Punnett square changes to this. And then we end up with these potential offspring. Right? Correct. Okay, and so there is our F2 or our second filial generation. And what is the genotypic ratio of these individuals? Say it louder. I'm sad. 1 to 2 to 1. All right, so genotypically is 1 to 2 to 1, and we always represent it as homozygote dominant, heterozygote, homozygote recessive. Right? So we have 1 homozygous dominant, 2 heterozygotes, and 1 homozygous recessive. So 1 to 2 to 1. Phenotypic ratio. Phone is going crazy. Phenotype. Oops. Phenotype. Yeah. Spell better than me. 3 to 1. Right? Because when we're talking about phenotype, we're looking at whether they're tall or short and what ratio. So tall, tall, tall. Okay. All right. So, and of course, yeah, these next couple of slides are just sort of walking us through. And I'm gonna wrap up here. So actually, this process, the monohybrid crosses, are what allowed Mendel to make his first law, which was the law of segregation, or the principle of segregation. This is literally what it states. Each individual diploid organism possesses 2 alleles for any particular traits. True? Yes, true. Those 2 alleles segregate when gametes are formed, and 1 allele goes into each gamete. All right. And then our dominance principle, which we've already talked about. I'm sure that you are tired of me saying things to you and then repeating them over and over again, but I'm hoping that you remember them. It's good pedagogical process. So, okay. And here, I'm sure people were still writing, which everybody's still writing. Continue writing. And then I think I'll give the last 15 minutes to work on problems, ask questions. And then on Monday, we will go through dihybrid crosses and look at pedigrees. So the worksheet for chapter 3 is looking at sort of 3 major concepts. One is our law of segregation, one is going to be the law of independent assortment, and then the third will be, I think there's like 1 pedigree practice problem on there. So you can go ahead and start. How many, I don't want to ask that. I'm assuming that most people at this point have the textbook. I would encourage you to be at least skimming it or reading it before you come to. Yeah, I know the answer is like maybe 2 people in the whole class will actually read the textbook. I too once did not read the textbooks. Just being honest. All right, so just quickly, this is walking us through another example, and so just again to go through it quickly, we have 2 different seed types, round and wrinkled. Remember, these are always going to start with a parental generation that's true breeding. We cross those 2 and get our F1. Our F1, as long as we're looking at 1 trait that's true breeding, is going to look identical. Every single F1 offspring is going to have the same genotype and the same phenotype just by default. We then self-fertilize, and sort of here are actual results that Mendel got. So he counted in the F2 generation 5,474 round seeds and 1,850 wrinkled seeds. How many people want Mendel's job? He counted tens of thousands of seeds probably every growing season. That sounds horrifying. And so this is sort of his outcome, right? And you take those and you put them into a proportion or ratio, about 3 to 1 round to wrinkled. Okay. And so he concluded that this follows the law of segregation, and we can show that with our work. And this is just more of the same. All right, And so this is just a table that wraps up Mendel's, all of his experiments, so all of his traits for the monohybrid. And you can see, and we'll talk about this on Monday, are any of these perfectly 3-to-1? He got pretty darn close here, right? But he also counted 8,000 seeds for this particular trait. These others are a little bit off. We'll talk, and I've already mentioned this a little bit earlier this week, but we'll see how we can actually take these values and determine if they are consistent with the 3-to-1 theory or if they are too far away and something else is going on genetically in these plants. Oh man, I really wanted to learn this. Okay, so awesome. We will, I told you, repetition. Okay, so take the next 15 minutes, 13 minutes, and get out your worksheets, ask me questions about those if you have them. If you have questions about something that you want me to go over, I'm happy to do that too. If you want to start working on the next worksheet, that is also an option. But take advantage of the while I'm around.